Hi everyone, thank you for coming to this webinar. I'm Naoto Kroda, the moderator of today's webinar. I'm a board-certified neurosurgeon in Japan and currently working as a research fellow at the Wayne State University, the state of Michigan in the US. I'm also the leader of Research and Translational Task Force in YesILAE. Today's webinar topic is EEG as a tool to dissect epileptic network. Today, we have two speakers in this session. The first speaker is Dr. Mark Cunningham from Discipline of Physiology, School of Medicine, Trinity College, Dublin in Ireland. The second speaker is Dr. Julia Jacobs from Alberta Children's Hospital, University of Calgary in Canada. Both speakers are well-known experts in the field of epilepsy and electrophysiology. In this session, firstly, each speaker will have a presentation about this topic. After the two presentations, we will have a Q&A session or interactive discussion session. Even during the presentation, the audience can type and post their question to the speaker on chat. And during the Q&A session, I will ask the audience question to the speakers. Okay, then let's get started. Again, the first speaker is Dr. Mark Cunningham. Dr. Cunningham, could you please share your screen and start your presentation? I think you are muted.
Okay, thank you very much. Can everybody see my screen okay?
Yes.
And hear me okay? That's great. So thanks very much, first of all, to the organizers of this and for the introduction from our host today. And it's a pleasure to be able to speak to you today from Dublin. Good afternoon or good morning, depending on what time zone you're in. My task today is to discuss with you from a preclinical perspective, why do we need to record EEG in animal models, and really what is the benefit of using EEG as a tool to dissect epileptic networks. So I just want to start off by giving a few learning objectives for this session, which I always think is very useful. So I think what we're trying to achieve today is really to understand the need for EEG recordings in animal models of epilepsy, to understand the different methodologies that can be used in terms of EEG approaches in basic research, basic epilepsy research, preclinical research, and then to also understand some of the limitations of EEG approaches that are used in basic epilepsy research. So first of all, what I want to do is kind of really set the scene and ask the question of why do we need EEG recordings in animal models of epilepsy? And you're going to hear a lot more from our second speaker today, Dr. Jacobs, about human EEG. But we are all pretty well aware that, EEG is a gold standard in terms of one of the diagnostic tools that's available to clinicians in terms of being able to non-invasively monitor the brain in some cases, also invasively in some cases as well, but to be able to capture the electrophysiological or neurophysiological state of the brain and to be able to localize regions that are generating aberrant electrical activity. But what we also now are becoming aware is that the other power of EEG, particularly with high density recordings and digital recording techniques, is that it allows experimentalists and clinicians to look at distinct patterns of activity. For example, as shown here in the middle panel, high frequency oscillations, which could be potential biomarkers that give even better information to clinicians to aid in that process of localization of a seizure focus. Now, a lot of what I'm going to talk about in the session today, obviously, is what we try to do in our preclinical animal models. And again, a lot of that is done in rodents, using rodents. And what's reassuring is that, of course, a lot of the architecture is preserved in these rodent brains that in some way captures the human brain. And it's also very reassuring that a number of these signals, these distinct signals, such as high frequency oscillations, for example, are also recapitulated in these rodent models. So it's worth just also mentioning that we also have a range of other preclinical animal models that are available to us. And of course, the strength of these preclinical animal models is that we can also combine electrophysiological or neurophysiological recording techniques with monitoring of behavior. So we can correlate with behavior using animal models. We can look at seizure susceptibility. We can look at the changes that take place from an EEG perspective with respect to epileptogenesis. And as you'll see later on in my presentation today, we also, because we have this experimental model, are able to then examine the efficacy of new and emerging anti-seizure medications and indeed novel therapies that are now coming online. And I'll talk later on about one of those specifically, which are gene therapy approaches. So again, you can see here the range of animal models available to us, and in all of these, EEG can be employed. So we can go from a rat or a mouse up to a complex species such as non-human primate. And again, there are very good genetic animal models that straddle all of these species. But we can also induce seizures in normal animal models using, for example, chemoconvulsant animal model approaches. So what are the EEG approaches that are used in basic epilepsy research? So there are, generally speaking, three approaches that dominate. And again, these vary in terms of the type of recording and the complexity of the signal that's being captured, both in terms of the spatial domain, but also the temporal domain as well. So the most straightforward technique is known as the surface EEG approach, which is a single screw electrode. So an electrode, screw electrode is placed in the skull. And this is analogous to the human ECOG approach, the electrocardiography approach. We can then move to a much more sophisticated approach, which is intracortical EEG. This is using cutting-edge technology, multi-electrode arrays, where now what we can capture is not only the LFP, the local field potential, which is the underlying signal that generates the EEG, but we can also now start to discriminate down to the level of individual neurons by discriminating single units. And the analogous scenario here in terms of human EEG are human depth electrode recordings that are conducted by a number of epilepsy surgery centers around the world. And then finally, we have a subdural EEG, which is a flexible array. Again, these are large electrodes, but we now are able to record over areas of the cortex, and again, capturing the local field potential. And again, we know that this is a similar technology is deployed in human ECOG recordings, particularly subdural grids that are used in epilepsy surgery programs. So these are the kind of common three technologies that are used. And what I'm now going to do is I'm going to try and give you some case studies that actually demonstrate how these techniques are used in basic epilepsy research. So I told you earlier on about one of the strengths here is that using preclinical animal models and deploying EEG technology, we can, of course, bring to bear novel experimental approaches to try and see whether we can get better therapeutic interventions in our animal models of epilepsy. And this is one example where this is work that has emerged from the epilepsy research group at Queen Square at University College London, where the problem is that there is a brain insult which causes trauma. There then is the construction of an epileptogenic network following a latent period, and then we have an epileptogenic network which causes an epileptic seizure as seen in the EEG trace. Now, of course, the therapeutic strategies that we have at the minute is that we can use anti-seizure or anti-epileptic drugs. Of course, the difficulty there is that we still have to contest with a 30% of patients which demonstrate drug-resistant epilepsy. And of course, there are comorbidities associated with the epilepsy, and there are also other side effects of the drugs that are deployed as well. So what's emerged over the last number of years is the thought that much more sophisticated approaches could be deployed, and in particular using gene therapy approaches. So here what we can see over on the right-hand side is this particular approach involves the deployment of a virus with a gene for a particular potassium conductance, the Kv1.1 channel, and this is expressed in excitatory principal neurons. These are neurons that are driving the epileptic discharges in the neocortex. And the aim here is that by essentially expressing this increased potassium conductance in these excitatory cells, it will dampen down cellular excitability, and that this should essentially either block epileptogenesis or indeed restore normal activity. And so the panel on the top right-hand side shows some of the experimental data here, where we can see a pyramidal cell that's been recorded from using whole cell patch clamp technology. And this is a control pyramidal neuron. And then in the yellow trace is a similar neuron which has been transfected with the Kv1.1 channel. And you can see this very clear reduction in the frequency of action potentials, which suggests that there's a dampening down of excitation. So what we can see here is that at a cellular level, this technology is clearly able to limit excessive excitability in cortical pyramidal neurons. But what does that mean in terms of how cortical networks generate seizure activity? And this is where the EEG can be combined with this is a model that's been around for many years. This is the tetanus toxin model, which essentially involves the injection of tetanus toxin into an area of the neocortex, and that creates an epileptic foci. So it's a very good model of focal epilepsy, and you can see that you get this refractory focal neocortical activity, which can be picked up on the EEG recordings. So what the group in London did was they did a very beautiful study where they actually used some tricks from clinical trial processes. So they did this preclinical study using randomized control trial approaches. They had two groups, one that received the virus and one that received just a control, a green fluorescent protein with the virus. And they essentially then implanted the screw electrode to allow them to monitor continuously the ECOG, the EEG, in animals that had either been assigned to the control virus injection group or the group that had been injected with the virus and the associated genetically modified potassium conductance. And you can see here, they then recorded over a month-long period. They also were able to do simultaneous video recordings to look at the behavioral phenotype. But here in this slide, what we see is a quantification of some of the ECOG data that's been recorded from these two groups in terms of the treatments that they've been ascribed to. So the first thing to see is that in terms of seizures relative to baseline, you can see that there's a marked difference in terms of the number of the seizures in the control and in the group that had been injected with the modified potassium conductance. We can also see a very clear difference in terms of the development of the cumulative seizures over time in the control in the recording period. And we can also see a significant reduction in terms of post-treatment seizures. So this provides very good evidence that you can see the strength of being able to combine this EEG approach in a preclinical model using a novel gene therapy approach. Now, of course, one of the limitations of the EEG approach is that whilst it's excellent at being able to demonstrate what's going on at the macro level, so in terms of networks within the brain, as I've shown in this case study, the difficulty is that it's very hard to capture some aspects of what's happening at the microcircuit level. And for example, the previous case study was a good example of where we can see the deployment of trying to understand features of the microcircuit using wholesale patch clamp technology to demonstrate that the gene therapy approaches are working, and then translating that to the macro using the surface EEG recording techniques. But can we go the other way? Can we go from macro to micro, or maybe try and combine some features of that? And the reason for this is that there is really a discrepancy between neuronal firing patterns and the EEG signal. So it's worth emphasizing that in regions that are implicated in epilepsy, such as the cortex and the hippocampus, we have neuronal microcircuits which show remarkable heterogeneity in terms of both the cellular and synaptic aspects. And the central tenet derived from EEG is that epileptiform activity equals recurrent and hypersynchronous activity. But on closer examination, however, what's emerged is that neuronal activity during seizures and epileptiform events is actually highly heterogeneous. And there's evidence, for example, from this study from Truccolo et al. In 2011 that shows that there's a real disparity in terms of when you start to actually monitor very large numbers of neurons. So here what you can see in this trace is you've got the local field potential recording here at the top, the associated spectrogram, and here you have a raster plot of probably nearly 150 neurons. And you can clearly see that some of these neurons are, yes, they are clearly participating very much during the seizure events, but others are clearly not. So these new approaches using NeuroPixel and other multi-neuron patch clamp techniques are going to provide us more information about the complexity of how we can essentially resolve what's occurring at the level of individual cells with the EEG signal. So the second case study that I want to demonstrate to you is another type of approach that's used in basic epilepsy research, which again combines some of these electrophysiological approaches. So again, the signals that underlie the EEG, the local field potential, but attempts to get a little bit closer to the patient. And this is an approach where preclinical scientists can access human resected material from patients undergoing neurosurgery for refractory epilepsy. And then this tissue can be kept alive and it can be used to interrogate both the network and cellular features of these epileptic networks in vitro. So this is some data from my own lab where we were able to obtain material from patients undergoing neurosurgery for drug-resistant temporal lobe epilepsy. And what we were able to do was we were able to record the local field potential signal. So again, this is a signal that underlies the EEG. And we were able to demonstrate that we could see spontaneous epileptiform activity in these samples that we'd obtained from these patients. And indeed, when we looked back at some of the in vivo recordings that had been done in these patients, this is an in vivo foramen ovale electrode, we could see that the type of epileptiform activity, these interictal spikes, was markedly similar to what we were seeing at an in vitro level. And indeed, again, I come back to the high frequency oscillations, we could see the existence of these high frequency oscillations both at an in vivo and an in vitro level. Now, the strength of this approach is that in vitro, in these human epileptic brain slices, you can do your extracellular recordings. You can then conduct pharmacology, which is shown over here on the left-hand side of the panel. So you can ask questions to interrogate the pharmacological sensitivity of these networks. But the real strength comes when you combine extracellular recording techniques, which again are the correlate of the EEG, with intracellular techniques. So now we can record from individual neurons and try and correlate that behavior with what is occurring at the level of the network. And so we did a very detailed study to look at, for example, the degree of excitation and inhibition that various cells receive during this network activity to try and build up a picture of what was occurring within the epileptic network. And what we could also do then is we could then start to ask questions about very distinct cell types. For example, in these recordings, we were able to record from inhibitory interneurons, and we could then look at the relationship that was being taking place in terms of describing the features of the network, in particular the high frequency oscillations. And again, we were able to observe these spontaneous interictal sharp waves and the very fast oscillation. We were able to demonstrate an explicit relationship between non-synaptic activity and high frequency oscillations, and that was confirmed by pharmacological antagonism of gap junctions. And again, we could then say something about how interneurons, particular cell types, behave during this network activity. So just to conclude, I hope what I've been able to demonstrate in my presentation today is that EEG offers a key tool that allows us to understand translational biomarkers that are critical in epilepsy. It also allows efficacy testing for novel therapies, for example, gene therapy. And EEG is optimal really for macroscopic brain activity, but it doesn't capture or provide microscopic information. So again, novel approaches are coming online that will fill this important information gap. So again, I'd just like to thank the organizers and thank those of you that have signed in for this webinar today. And again, just to acknowledge our funders, SFI, and I'm part of the Future Neuro Group that's based here in Dublin as well. So thank you very much, and I'll be happy to take any questions.
Thank you for your great presentation about animal models of EEG to understand the epilepsy. Thank you. It's very structured and informative. We will have a Q&A session after both presentation. So let's get to the next presenter. The second speaker is Dr. Julia Jacobs. Dr. Jacobs, could you please share your screen and start your presentation?
Yes, can you see my screen well?
Yes.
Wonderful. So then I'm going to get started. Just as some words of background to my presentation. So this is, as you probably could see from Mark's presentation, a follow-up from a session that we had done at the conference in Dublin. And my task at this point was really to see or to show, and that's what I'm going to do again, how we can combine our knowledge that we gain from recording EEGs in the animals and translate it towards human EEG and knowledge about challenges in human epilepsy. And to start off, my slides are moving. I want to give you a little bit of a riddle and a task, and everyone can either answer it for himself or go ahead and post it in the chat. I've posted 2 EEGs here for you, and there's no secret about one of them is a rodent EEG and one of them is a human EEG. And I want you to take a guess on which of the 2 you think is which. And of course, the main take-home message from this is that human and animal EEGs, if you look at them in the same time scales, look very similar. I don't want to say that our brains function similar, but it's just the pure EEG traces and how we describe them can be very similar. And I think this is, I will give you a solution to this riddle at the end of my presentation today. So take a good guess. I think the reason why we often have the feeling that animal EEG is actually different, or at least that was my perception as a physician scientist looking at animal data, from the human EEG is because traditionally we have developed different ways of presenting our EEG traces. So this is, I just chose 2 random publications to make my point. What you can see here is, for example, a recording of an interictal transition in a rodent model. And you can see here, if you look at the time scale, that this is very compressed, like this time scale is 1 minute. In human EEG, we usually have a much more extended timeline, so seizures and spikes and interictal activity look very different to the eye. And then if we move forward, many of the animal data is presented like this in very condensed versions. And then if we look at publications, the animal and the human EEG look very different because we usually don't show human EEG in this way. And I just want you to be aware of this difference. And sometimes I think, and this is why these kind of translational webinars are so important, we don't really talk in the same terms with each other when basic scientists and physicians exchange. And it's really important to understand which tools we use and how we describe certain matters to be able to communicate well with each other. The other thing which was striking to me when I did my first work with animals, and this is really me coming in as a physician and being asked to rate some EEGs and clinical scores, is how similar some of the seizures we see in animal models can look to human seizures. And I just randomly picked the temporal lobe seizures, and this is from the description of the Mayo Clinic's homepage about how a temporal lobe seizure looks in humans. And you can say we usually have like unawareness of the surrounding, staring, so behavioral arrest, lip smacking, repeated swallowing and chewing, and then finger movements or automatisms, as we call them. And then, of course, there's a possibility that this moves towards from focal seizure with impaired awareness to secondary bilateral generalized tonic-clonic seizure. So this is what we would see in the humans. And then this is from the Racine scale, which you're probably all aware of, where we scale temporal lobe epilepsy models in regard to their seizures. And if you compare, there's a lot of similarities. You have immobilization, frozen posture, facial automatisms, you have some forelimb movements. So I think there is a lot of similarities between the animal models and the human epilepsy. I also want to just give you a little bit of a hint for the rest of my talk. There will be 2 example pictures for what is animal data and what is human data. So you will find this little guy here whenever I'm referring to a human data study, and you will find this red as a representation for any animal models in the rest of my slides. Moving on, what do I think are the advantages of animal recordings? And I think Mark did already a marvelous job talking about this. So from a clinician's perspective, looking back and what I would want animal models for is really that we can do flexible long-term recordings. If you treat your animal well, it will not be angry if you look at it for weeks. That's very different in human epilepsy, where patients usually get upset with us if we keep them in the hospital for too long. We can create a standardized environment and standardized tests, which are not always possible in humans. We have what I call unlimited spatial and temporal resolution, because you can use as many electrodes as you want. You can try new electrodes which are not licensed yet. You can look with more or less spatial temporal resolution. And then last but not least, we have much more ways to look at tissue analysis, modulate tissue, look at histology, which is of course all not possible in the human. And you are all aware that we get, as a result of this, we get much more detailed recordings of subcategories of the hippocampus in animal models, and we can learn much more about mechanisms. But the challenge is when we use animal models to refer to human disease, that there is a big scaling difference. And this is from what we have learned about high frequency oscillations, and Mark has already mentioned these events. But what I think is quite important here is that a lot of the recordings which were early done in the mice model have been recorded with either microelectrodes alone or macro combined recordings. When we talk about human data, we are going on a completely different scale, looking at intracranial macroelectrodes, and we also now use a lot of scalp EEG recordings to look at certain biomarkers, especially also HFOs. So we have to be aware that we are translating from a different brain anatomy and with different methodologies when we translate animal data to human data. And this will be a bit of the part of my talk. How can we learn from animal models for clinical care, and how can clinical care inform research in animal models? And I want to use the high frequency oscillations as a biomarker example, but I think there's a lot of other diseases which you can use to do the same comparisons. Just as a quick reminder, when we talk about high frequency oscillations, we usually look at events on the upper frequency range. So this would be in the ripple range between 80 to 250 hertz, in the fast ripple range 250 to 500 hertz, and then in the very high frequency oscillations at around 1000 hertz. What do we know about high frequency oscillations and their importance for epilepsy? We have learned over time that we can either use high frequency oscillations as a localization tool. So whenever you want to know in your patient which part is epileptic and which part might have to be removed, for example, in pre-surgical settings. And we also know that they reflect to some extent the seizure propensity or the disease activity of our patients with epilepsy. So they can help us to predict the risk of recurrent seizures, risk of refractory epilepsy, and maybe the treatment response. And I'm going to talk a bit more in detail in the coming slides. So what the point I want to make today, or I want to discuss with you, and this is like the overview slide to my talk, I want to discuss with you for the following hypothesis. Is it true that linking results from animal models of epilepsies to finding in humans completes our understanding of neurophysiology in epilepsy? And I want to discuss 4 points about this. First, do animal models allow us to find new discoveries and observations, which we would not find in humans otherwise? Do they allow us to study epilepsy in a more standardized environment? Do they allow us better to separate physiological and epileptic activity? And last but not least, do we learn more about networks by looking at animal models? So let's start with new discoveries and observations. And I think for that topic, high frequency oscillations are really the best paradigm. Because we know of HFOs at all because of early animal studies, and it has been really Anatole Bregan from UCLA who has been spearheading this field now more than 25 years ago. First describing these events in a KINET AZ rat model. And what was really exciting, those of you which are familiar with this model, is the animals undergo a status epilepticus, and then not all of them, but a subgroup of them develops chronic epilepsy and chronic seizures. And what he could show for the first time is that there are these high frequency oscillations, in this case fast ripples, who only occurred in those animals who actually developed epilepsy, while those who stayed seizure-free after status did not have fast ripples. And this was the very first very clear link that fast ripples have to do with epilepsy. And then talking about translation, this first data was done in microelectrodes and animal models. And the group in Montreal, and at that time I was a postdoc there, then started looking at, do we see these same events in the humans with macroelectrodes? And it was first Elena Ostarathu who published on this in relation to intellectual spikes. And then we had this early publication where we linked human high-frequency oscillations recorded with macroelectrodes to the seizure onset zone in the human epileptic brain. And if you're wondering, this is how these events look like. So a classical example of how we find something in the animal model, then translate it into find it in the human, and then move on from there. At this point, we have done a substantial number of studies which try to link removing high-frequency oscillations during epilepsy surgery and whether this actually results in a seizure-free outcome for the patient. What you see here is an early meta-analysis that we did about this topic. Just of note, because I'm going to chat about this a little bit later, there is a difference. There is not really a difference in whether you look at ripples or fast ripples for this topic in the meta-analysis. And we have moved on to several studies, two of them trying to predict the post-surgical outcome by looking at the proportion of HFOs that was removed, and one of them actually doing a randomized controlled trial which compared the usefulness of ripples, fast ripples, and spikes in regard to predicting, to tailoring surgery, and this way improving post-surgical outcome for patients. So we have really used this into trying to move it towards clinical care, and I think that's quite exciting. But we also noticed some caveats by doing this, by moving from the animal to the human findings. And one of these caveats is that the separation between ripples and fast ripples, as I just mentioned, is more prominent in the animal model than it is actually in the humans, and we are until now not sure whether fast ripples are really more epileptic than ripples, and I'll discuss that a little bit later. We also learned that separating physiological and epileptic high-frequency activity is not as easy as we think, and definitely more difficult in human findings than in animals. And we also learned that the spatial extent of an event and the generator size might be very different between animal models and humans, and that can also affect how we can translate information from the animal to the human. Next, moving on to the next question, do animal models help us to study epilepsy in a more standardized environment? And I want to give you a couple of examples for this. So this is actually two studies that we did back to back when I was still working in Freiburg, looking at the evolution of high frequency oscillations during seizures, before and after as well, and whether there is a difference, whether in the beginning of a seizure, so not in the full-blown seizures, but directly at the seizure onset start, there is a difference in high frequency oscillations depending on how strong the seizure is. So do HFOs mirror seizure severity in individual seizures? And I've introduced you to the racine states earlier, and I think this is a very, that was an animal study where we looked whether there was a correlate between the number of high frequency oscillations and the racine state of the animals, and we separated the big racine 5 and 6, so big seizures, from the smaller focal seizures. And as you can see, we didn't see this in the pre-ictal phase, but there was a significant difference between the higher stages of racine and the lower stages, both during the seizure and actually in the post-ictal period. We then went ahead and said, if we can see this in the animal model, maybe we can also see this in the human temporal lobe seizures. And again, we looked at different periods, and this is now from the early seizure period, and you can see ripples here on the left and fast ripples on the right. We can focus here on the seizure onset areas for now. And what you see here is that we didn't separate racine states, of course, because they don't exist in the humans. But what we separated in blue is the seizures which started as temporal lobe seizures and stayed focal. So we don't have secondary bilateral generalization. And then the red ones are those which moved on to generalize. And you can see that there is significantly more HFOs in the early stage of a seizure if the seizure is tending to become generalized. So a nice work where we can see that there are similar findings in the animal model as in the human work. Talking about standardized environments, I think another way where we know this and where it's actually very helpful is looking at epileptogenesis. So does someone with a risk or a first incident develop chronic epilepsy, and in which timeline? And there has been early data, as I mentioned earlier already, by Anatole Bragan, who showed that if you develop chronic epilepsy after a first status, the timing of the HFO is linked to the timing of the first seizures, and it's also the occurrence rate is also correlating with the first seizures. So there was clear incidence that somehow the number of fast ripples reflected when animals are moving in their stage of epileptogenesis towards developing chronic epilepsy. The other thing where we can use standardized environments is to try to predict in a risk group which animals at risk or humans at risk will develop chronic epilepsy afterwards. And this is from a summary work that Mike Zielmans and I published a couple of years ago in Epilepsy Currents. And what you can see here, there were 2 studies in animals with traumatic brain injury, which showed that animals who developed seizures afterwards had early on repetitive high frequency oscillations. And then there is a study, which I will show you in detail in humans, so in children, where we actually showed that we can use HFOs to predict who will develop epilepsy after first seizure. Just to give you a little bit background, what we did in this study is that we saw patients in the first week after they had the first unprovoked seizures. And in children, only around 50 to 60% will actually develop seizures after they had a first epilepsy, after they had a first seizure. So it's actually a 50-50 chance of prediction. And we use these very first EEGs to look at ripples, spike ripples, and spikes alone, and predict whether 1 year later these kids had a diagnosis of epilepsy. And as you can see, the best separation between the epilepsy and the non-epilepsy group were actually the ripples and the spike ripples, while the spikes alone were absolutely zero helpful to predict whether someone will have a diagnosis of epilepsy or not. And this is a bit more detail. You can see that the ripples have the best ROC to predict, but they are more specific than they are sensitive, so you can also use the spike ripples together. And the exciting part about it is that this would be a biomarker where we can give parents who, of course, want to know, is this a single event or does my kid have an epilepsy diagnosis, a prediction early. Moving on to separating physiological from epileptic activity. This is a big challenge because the majority of the data that I've shown you, both in animal models and in humans, is from intracranial EEG recordings. And of course, it's absolutely unethical to record intracranial EEG from a healthy human brain. So we don't have a lot of data from that. And there would be a big hope that maybe using animal models will help us better to understand what is a physiological HFO and what is an epileptic HFO and what is really linked to epilepsy. And just to roll back a little bit, we've known for a long time that there's physiological high frequency activity, better known as, for example, the hippocampal sharp wave ripple, who was first introduced by data a long time ago from Buzaki's lab. And he has this nice overview presentation where he clearly shows that across different mammals, sharp wave ripples when they are linked to memory consolidation, actually look very similar in their EEG traces, as you can see here. So when Anatole Bracken looked at his first animal models, there was always the notion that we have physiological ripples in the hippocampus, and that when they turn to fast ripples, this is when they indicate epilepsy. And this is what you can see here from one of his publications, where he linked fast ripples in the animal brain very clearly to epileptic activity. And that was then translated by the group in UCLA, again, using microelectrode data into human recordings. And these were classically bilateral temporal implantations, where there was predominantly one hippocampus involved in seizure generation, but usually you have some involvement of both temporal lobes, because if you don't, you wouldn't implant the patient, but just go directly ahead with a resection. And what they found was that there is, the data suggested that there were physiological ripples in the data and epileptic fast ripples. This could not be confirmed with the bigger data sets that I showed you earlier on the surgical outcomes, for example. We could see that sometimes ripples and sometimes fast ripples were predictive of epileptic areas and surgical outcome in the human macroelectrode data. And this is actually a distribution of physiological HFO activity across the brain, published by Dr. Frauscher. And you can see that there's two challenges. First of all, physiological activity can be in both frequency ranges. And then also, as you can see, it's not limited to the hippocampal structures, but actually can be found all over the brain. All the blue areas are the ones where we have little physiological HFOs, and the yellow areas are those where we see a high amount. We also have data analysis, which actually from humans shows that there is not really a separation at 200 or 250 hertz for ripples versus fast ripples. So most of the events are mixed events, and it might be an artificial separation to talk about ripples and fast ripples. When we talk about physiological versus epileptic, we now know that this is much more complex. Separating epileptic from physiological HFOs might be possible by looking at interactions with epileptic spikes, association to sleep phenomena, and occurrence and predominance over certain brain areas. It is beyond the talk today to explain all the different ways that we have tried to separate the red, so the more epileptic HFOs, from the green, the more physiological HFOs. This is just a summary slide, which I wanted to use to show you that matters of HFO might be more complex in the human brain than the animal model has suggested before. And we always have to be aware it doesn't have to do only with the brain, but it might also be influenced by the different recording methods that we are using between animals and human recordings. And of course, with the different brain areas and brain functions that we can cover in humans, which are certainly different from an animal model or animal brain. Moving on to the last question of today, can it help us to understand network activity? What we knew from the very early studies in animals is that fast ripples in the animal model had a very strong spatial stability. So they stayed in the same area of sub-area of the hippocampus during the time of epilepsy. and they had very small focal generators. We now learn from human epilepsy that there is actually networks involved into HFO oscillations, that they are not as focal in their generators, and that they might propagate and have network interactions similar to what we know from epileptic spikes. So that's important to acknowledge, because it is a limitation that we can hardly study in the animal model, because brain networks in the animal are so different from human networks. But as I said already, ethically, it's a challenge to look at different time zones in this intracranial EEG. For example, you would never implant a patient again after epilepsy surgery unless he's really doing poorly. So we never record from someone who has had a good or successful epilepsy surgery, right? My group has tried to answer some of these questions by looking at a non-invasive way or minimally invasive way to treat patients, which is a radio frequency thermocoagulation. So that's a method where you use your implanted stereo EEGs to burn part of the brain away where we think that they are the seizure onset. And this is a good way to say there might be areas where I cannot study it with the animal model. So I have to find compromises how I can study this actually in a patient. And it was actually an interesting experiment for us to see that we could finally record from patients who had an intervention done where we manipulated the network and what we would see afterwards. The main findings of this study was that HFO reduction in one area, if you burn one area, goes way beyond the vicinity of the intervention. And there is a significant reduction in functionally connected areas of HFOs, even beyond the lobe of the intervention. And whenever this happened, this was correlated with a better seizure outcome after the intervention than if the local intervention did not have a widespread effect. Just as one example of sometimes when we have to work in the human EEG to get our questions answered. To conclude from my talk, EEGs and seizures in animals and humans are often more common, have often more in common than we think. EEG in rodents has led to the recovery of HFO, which is a critical biomarker for human epilepsy at this point. Often we learn by looking at both sides, find similarities and find differences. And for ethical reasons, some questions cannot be studied in the human brain. And for this, animal models are really the key. I also want to note, finish on another note, which is that we just now had a very interesting publication about HFOs in Alzheimer's disease in animal models. So I think, again, animal models can really drive us with ideas. And I think this is probably the next thing which also will be studied in humans to see whether we can find this exciting finding, very similar to 20 years ago when Bregan described this the first time in epilepsy or animal models of epilepsy. I promised you a solution to the quiz. So the colorful EEG is the animal model EEG. The black and white one is the human EEG. I'm curious to hear in the chat how many of you guessed that right, because I think from just looking at it, it's really difficult to know. Last but not least, I wanted to let you all know that we have this exciting workshop coming up. There's still spaces available if you want to sign up and if you want to travel to New York. It will be focused on high frequency oscillations in epilepsy. And with this, I would like to thank you for your attention. I would like to thank all the funders of my different studies and of course my research and clinical team I'm working with, both at the University of Calgary and the University of Freiburg. And if you want to learn more about epilepsy and EEG and HFO, follow us on social media at Excited Brains. Thank you.
Thank you very much for your presentation. We can understand why animal studies are important and what animal models allow us to do from point of view of clinical investigator. Thank you. Okay, then let's go to the Q&A session. We have a lot of questions from audience. Let's get from the first one. First one is the question about expected or potential side effect in vivo about channel therapy. So this question is, I think, about Dr. Cunningham. With the modified KV 1.1 channel during the whole cell patch clamp, seizure and excitability reduced, but is there any expected side effect?
So, so the work done so far in the animal models, I mean, I didn't have the time to show this data, has demonstrated that the deployment of this gene therapy approach doesn't modify normal behavior. So they've looked at a number of kind of standard rodent behavioral assays, you know, Y-maze, other types of behavioral assays, and certainly it doesn't cause any effect. And in fact, in more recent work, so they've actually now used the CRISPR-based upregulation of the KV 1.1 channel in an animal model of temporal lobe epilepsy, and they demonstrated that that actually rescued the cognitive impairment that was associated with chronic epilepsy. So it suggests that certainly it's not having any effect on kind of normal background behavior, but in the animal models where there are cognitive impairments, it seems to be normalizing that impairment. So you're kind of getting this double effect in terms of reducing the seizures, but also helping to treat the cognitive issues associated with epilepsy. So that's quite promising in that regard.
Thank you. Okay. Let's go to the next question. Next question is, I think, to both of the speakers. This question is about the mechanism or origin of physiological HFO. So physiologically, what is the origin of HFOs? What cell groups are capable of generating such rapid oscillations, and why it's so little, very short time? Do you have any idea or comments in like animal study size or human study side?
Maybe I can sort of take that one first, Julia. I mean, there are a number of hypotheses out there, and I mean, I have to be sort of equitable in terms of giving time to both of them. So there's been one idea that that these high frequency oscillations may be a manifestation of GABAergic inhibition, because we know, for example, when we think about how cells generate action potentials in the central nervous system, that for example, cortical inhibitory interneurons are very good at being able to generate action potentials at very high frequencies. So, for example, the parvalbumin containing inhibitory interneuron, which is sometimes known as a fast spiking interneuron, can generate action potentials up to frequencies of hundreds of hertz. So that would certainly match with the kind of the temporal aspect of the high frequency oscillations. However, what has also been shown is that these high frequency oscillations can continue to exist in the absence of GABAergic mechanisms. So, for example, pharmacologically, you can block GABA A receptors and you can still get this type of activity. And so an alternative hypothesis that has been put out there by Roger Traub and others is that actually these high frequency oscillations are generated by a network of cells that are electrically coupled through gap junctions, and that this allows this kind of network type activity in axons of principal cells, that then can generate these high frequency oscillations. There's also another theory which has been put forward by researchers in Spain, that it actually could be the pyramidal cells, but that essentially, whilst the output of pyramidal cells is not at the frequencies, you could essentially have some kind of concatenation of multiple pyramidal cells ensembles that are then producing this type of global network activity that's seen at these high frequency oscillations.
Thank you. Is there any comment from human studies side, Dr. Jacobs?
You maybe have to move on because there's lots of questions and I think it was covered really well.
Thank you. Okay, and other questions is also related to the physiological HFO. One is what do you think about the event related to HFOs, and other is effect of pharmacological therapy on HFOs. Is there any comment?
So I think what I couldn't go into detail is that the most promising way of separating physiological from epileptic events is probably one approach is to use these systematic kind of atlases where we know that certain brain regions are more prone to have physiological HFOs, but this leaves you with a problem on how to separate physiological from epileptic in these high active areas. So for example, the mesotemporal lobe has a lot of physiological HFOs as well. And these structures, I think it can be super helpful to correlate with different types of events. So far, I think we have no indication that suggests that an HFO linked and coupled to an epileptic spike would be physiological. So that's definitely helpful to look at. There's also been ways to look at different sleep events and how they link to HFOs. And for example, if you have a HFO coupled to a sleep spindle, then you are more likely to, or then this is more likely to be a physiological HFO. So you can also do systematic sleep stage analysis to separate a physiological from epileptic events in the mesotemporal structures. And there's also some data that shows if you look at the variability of features within HFOs within one region, the more monomorphic the HFOs appear and length and frequency and amplitude, the more likely they are actually epileptic. So there's different ways to separate this, and it's definitely probably the most promising way to separate. Coming to the second question, there is good data which shows now first the intracranial and now in the scalp EEG that if you successfully treat someone with the medications, and that's independent of which medication, your high frequency oscillations will go down. And actually they are better at measuring this than the epileptic spikes, because even if most people believe that they treat the EEG, the epileptic spikes become less. In many childhood diseases, for example, this is not the case. But the HFOs are very distinctly going down if you treat successfully, so that's promising. And again, matching with the topic of my talk, this is actually very closely reflected in animal models because we have some studies for Keppra and lacosamide in animal models which show that HFOs go down whenever you treat the animal. So again, this is data which is informed from both sides.
Thank you. Actually, did you consider a physiological reports in your paper about the predictive value of scalp EEG, scalp EEG report to predict the epilepsy after the first seizure?
No, because this was actually, we didn't see a lot of physiological, on the scalp, you see a lot less physiological activity, and this is scalp EEG data, so it was much easier to separate. And our thought is that probably the majority of the events we saw were epileptic because they were so linked to outcome. But we are currently, actually, this is one of my studies, trying to design a scalp physiological event atlas over time of development, because we don't know whether kids might have more or less physiological HFOs on their scalp EEG than adults. So we are currently looking how this changes while the brain develops.
Okay, thank you. And the other question is about the methodology of automatically detect or analyze HFO. I guess there are many automatic detector of HFO. So is there any suggestion or comment from both sides?
Well, I think one important comment is that we just presented at last AES a comparison of 20 detectors on our intracranial data, and there was a lot of discussion around this topic. I think what is really important is you can use a published detector. Many of them are really good, but you have to adjust them and test them on your own data, because how well something will work is largely dependent on your artifact level, your recording equipment. So just downloading something from the internet and throwing it on your data is not a good idea. When we talk about scalp EEG, that's really challenging. Most of the groups at this point use a combination of automatic detection and visual verification to ensure that they don't detect a lot of artifacts.
Thank you. Is there any comment, Dr. Cunningham, about automatic detector?
I mean, I think from from a preclinical point of view, I mean, I think from in vitro studies, it's probably a little easier because we don't have the kind of issues that are associated with artifacts. Obviously, when you move to in vivo animal models, it probably starts to become more complicated, like humans, because you have artifacts. But I would just kind of again echo Julia's comments about making sure that the approach that you use is kind of well controlled for, and there's a variety of techniques that are available now out in the literature there.
Thank you. And regarding the artifact in the animal model, the one question is coming. Do you know of any good instrument or implants or system of recording EEG in freely moving mice?
So, I mean, there's a number of, and I'm not gonna plug any one system, but there are a number of very good systems now that you can get that again. I think it very much depends on the particular type of study that you're trying to do. But there are great systems now that, as I showed in one of the case studies, the gene therapy case studies, allow you to do wireless recordings. So the advantage there is that the animals are not tethered, which introduces its own problems as well. And with those wireless systems, what you can do also is it can be coupled to video recording. So essentially, it's like the video telemetry system that one might use in a human patient, but deployed in an animal model. So again, there are a number of both commercially available systems and other bespoke systems out there that again, the papers that I showed today have used to do these types of studies.
Thank you. And another methodology question is how trace EEG signals to different types of neurons? Are there any tools commercially available?
I mean, I'm not really sure what they mean by that question. I mean, I think obviously the EEG signal is derived from the LFP, and of course from that LFP signal, if you record with particular types of electrodes, you can record in a certain way. So very high digitization rates, 30 kilohertz. With micro electrodes, yes, you can pull out different types of neurons, but then you have to use sophisticated approaches like principal component analysis to essentially spike sort. And then what you can do is you can essentially cluster spikes based on the shape of the extracellular action potential to make some comment about, for example, this particular cell in this cluster here may be a putative interneuron, and these cells over here in this other cluster may be a putative pyramidal cell. And again, there are a number of tools that are available out there to do that kind of spike sorting, and to do it in a way that is, you know, robust, reproducible and reliable as well.
Thank you. And a lot of question is coming, so let's go like quickly. There are different mechanisms underlying the fast ripple and the very fast ripple. So different mechanism between fast ripple and very fast ripple. Is there any comment from?
Well, I don't think we can cover this all, but there is a lot of data on mechanisms. One of the papers I cited in my presentation, if you're talking about epileptic ripples and fast ripples, right? Sharp wave ripples, the one which are related to memory consolidation, are completely different. And I think some of the stuff Mark has covered in his question about mechanisms for fast ripples.
Thank you. And another one is, in physiological sharp waves ripple in hippocampal, neurons are supposed to fire in a specific order. Is it still the case in temporal lobe epilepsy?
I think there's been a paper recently that's demonstrated that phasing of sorry, I'm just trying to find the question here. That phasing of that sequence of spiking is actually disrupted, because of course we know that there are a number of that particular spikes, that sequence of spikes is known to be very important for a number of cognitive functions that are derived from the hippocampal networks, and of course that we see those being usually impaired in patients with temporal lobe epilepsy. So I think there's a preclinical animal model that described that in a number of years ago.
Thank you. And another question about the temporal lobe epilepsy is, what is the source or generator of bitemporal independent interictal spikes in ictally localized hippocampal sclerosis from animal model perspective?
I'm going to have to say I don't have an answer to that.
Okay. Thank you. We had a lot of questions, so sorry for over the 1 hour, but I hope this is a very informative and very meaningful for all of the audience. I thank for both of the speakers and audiences, and also the organizing committee of ESILA. It's time to finish, wrap it up. So thank you for Dr. Julia Jacobs and Mark Cunningham.
Thank you very much.
Thank you so much.
Bye-bye.
Bye. Bye-bye.